so far has been really unnoticed because of the technology. So my presentation will be split in 3 parts. A very short introduction on how Oxford Nanopore technology works. And then I'm going to talk to you about 2 things that I'm really passionate about. First, understanding large cohorts, in particular, large cohort of non-European population, and then how Nanopore can help you catching the variant that have so far not been noticed, in particular in rare disease. So Nanopore, Oxford Nanopore technology offers an end-to-end sequencing solution. It's very flexible, and you will have control at every stage. So the split is exactly the same than in any other platform, only it's all managed by Oxford Nanopore. So we offer library preparation. They are output optimized. They also optimize for speed. So we have solution for rapid sequencing, solution for more standard sequencing. And all our library preparation can be used either manually or can be automated on instruments such as Attica. What's really interesting, obviously, the sequencing. Our instrument can be either field devices, so the very small one like the MinION that you're most likely very familiar with, and larger lab device. When I say larger, they're never big. The biggest instruments is the size of an old-fashioned computer column, if you want. But that allows us to have a very broad range of output from very low to very high. And also being very flexible when it comes to price per gigabytes of data. Obviously, what's really important is also the analysis, and we work very closely with the community to provide solutions that are very accessible and scalable, but also very versatile. So we have a very active community of Nanopore users that creates tools constantly for new applications. And very quickly, so how the sequencing actually work. So when you do your library prep, you put a motor enzyme, and that's the little protein there in purple on the video. The motor enzyme brings the DNA in proximity to the pore, and it pushes the pore with very, very regular speed through the pore. Sorry, it pushes the DNA at a regular speed through the pore. And that allows us to have a direct read of the DNA or the RNA. The nucleotides have different current attached to them, and the pore read that electrical signal, and it turns it into a base call. So what does that allow you to do? It allows you to do a direct sequencing of either DNA or RNA, which gives you a very rich information because it gives you all the epigenetic information as you read them. Our library prep are PCR-free, so you don't have amplification bias. And we can come back on that in the next slide. We're also very read length agnostic. So a lot of people think we are a long read company, but actually the pore will read whatever you put through the pore. So if you put something really long, 4 meg, it will read 4 meg. If you put something very short, 700 base pair, it'll read 700 base pair, or even shorter. What's really important is that we provide real-time analysis. So on our PromethION, the base calling is done on the instrument as you do the sequencing. It's really important for some technology that are only proprietary to Nanopore or method, like adaptive sampling. And our chemistry is based on electronic that we're generating ourselves, manufacturing. It's very scalable from very small to very large, and it can be also really low cost. And you might say, okay, well, what does it mean for me? I'm a user. I don't really care about how the instrument works. I want to know what I can do with. Well, you can do with everything that's on that slide. So I'm going to go over it fairly quickly. By genotyping, I mean more targeted sequencing and not genotyping as you would do for genotyping on a microarray, for example. However, if you're really interested in really low pass application, you can come to us and we can discuss. But by genotyping, by targeted sequencing, I mean the thing that you're really used to already. So amplicon sequencing, enrichment chemistry, like pull down with twist and things like that, we are compatible. Again, we also have our own proprietary approach for targeted sequencing, which is adaptive sampling. I'm not going to go over it. If you have any question, we can go toward during the FAQ. And then there's sequencing. So if you're going to do whole genome sequencing, we have more or less two options. One's low pass, and low pass is not 1 to 2-fold. Low pass for us is 10 to 20-fold. But what does that mean? It means that if you put 2 samples per flow cell on a PromethION flow cell, you can still capture SNPs, methylation, structural variation very accurately for research purposes. So if you have a lot of sample and you're not looking for very rare SNPs or very rare variant, then that's definitely allows you to do more sample and have more genomic diversity at a slightly lower coverage. However, if you are more looking at rare variant, or if you're trying to work in a clinical setting, we highly recommend 1 sample per PromethION flow cell, and that should give you at least 30-fold coverage. And then when it comes to the application that are more around assembly, we've got 2 approaches. One that use ultra long reads, and it's done on a normal flow cell, but we recommend using 1 sample of a 2 flow cell. And you will all have heard about the T2T assembly because the T2T consortium released their publication in March 2022. And that's a very multi-mode approach. So it's a little bit more complex. We're combining very specific type of flow cell that are called duplex. And that's the only real case for which we recommend using duplex. And then we combine that with simplex flow cell for ultra long and some porosity. Again, if you have questions, for the sake of time, we can go over that during the Q&A. Because I really want to go into the meat of the discussion. So for about 20 years, 25 years, there's been a lot of genomic, a lot of GWAS. So there are a lot of genome-wide association studies. There have been hundreds of thousands samples that have been genotyped. And on that slide here, you can see there's at least 70 cohorts that have more than 100,000 samples per cohort. It's more than 50 million participants. There's 40 regions that have been represented. So you would think we have now captured most of the genomic diversity and all of the variant. And it's not the case at all. All of those studies, because they've been done mostly on genotyping microarrays and to some level a little bit on short-read whole genome sequencing, they are really focused on small variant and indels. And they are well characterized, but you've been missing, the whole community has been missing a lot of information, in particular on the large and more complex variant. Epigenetic EWAS is coming up a little bit, but it's not that big. Again, there are regions of the genome that are medically important, that are part of what is called the dark genome, either because the short-read technology or the array technology has not been able to capture it, or because the data analysis, because the regions are very repetitive, the data analysis is very complex, and the short-read do not span the region well enough to be able to align. And that's really been a massive miss for the for the community. So we're radically improving what we know today. The Telomere to Telomere Consortium published their paper in March 2022 to show that was for the first time they completed the sequencing of isomorphic chromosome. So one, you don't have 2 allele, you have 1 allele with long read end to end. Since then, they've shown that ONT is the only platform that can do telomere to telomere on a single platform. the effort of the telomere to telomere have moved on toward doing a much more representative analysis of the landscape, the genomic landscape of different population through the human pangenome. All those genome are phased, so they are not a collapsed linear genome like we're used to analyze. They are a graph genome, and they are realizing that it really increases the gene and the mutation conversion to disorder. And even the Y chromosome is completely sequenced now thanks to nanopore long-read sequencing. And in the word of Michael Atwaters, the use of a reference genome that is more representative of the population of a given patient will be more likely to accurately link the genomic variant and the disease. So we're really moving toward true genome reference. So again, those genomic insight really drive new biological discovery. So when it comes to dark gene, for example, and by that we mean the part of the genome either that can't be captured by physically by short-read sequencing technology or microarray, or you can add the sequence, but you can't align the sequence because they're too repetitive and there isn't a good reference genome. So the group from Mark Herbert has identified there's almost 3,000 dark coding regions across 748 proteins that are not well covered by short-read technology. It's quite important because it's at least 76 genes associated with over 300 human disease. And those are systematically camouflaged in plain sight. The other thing that's really important is structural variant. So 34% of all disease-causing variation are made of more than one single base pair substitution, so more than a SNP. And the structural variant contribute a lot more to the influence on gene expression than the SNP. So SV is more likely to have an effect on gene expression, up to 34 more likely to have an effect on gene expression than a SNP. You might ask, who's doing that? And who's doing that at scale? Who's actually doing long-read Oxford Nanopore technology on 100,000 of samples? So I've got two examples here that are quite recent. Genomics England, which is a national project in England that is actually used to help identify and characterize people with a rare disease and some cancer. I've announced that they will do 7,500 samples from their 100,000 genome project that have not been resolved at the moment with short-read whole genome technology. And another one in the UK that's really large is a large biobank managed by the NIHR, bioresource, of 22,000 samples, and those are more common complex disorders. So a lot of those are psychiatric disorder sample from people with a psychiatric disorder. And I mean, I can't miss the elephant in the room. You guys are in Malaysia, and I'm almost sure you're saying it doesn't really matter. All of that has been done on European participants, and it's true. And it's really a shame that 80% of the reference data are from or available data are from European participants. And there's a lot of need for more diverse data from a lot more population. A lot of those data have been generated by microarray using GWAS because it allows you to do a lot of sample. But one thing that's really important is if you guys are moving from microarray to contemplating doing all of your sample or some of your sample on sequencing, remember that about 8% of the genome is missed by short-read technology. That's a lot. 8% of the genome is the size of chromosome 1. It would be like you're not sequencing chromosome 1 at all. That's the biggest chromosome. So we do need to have data sets that are more representative of the population, but we also need data sets that are more representative of the complete genome. And that's why we're working with the 1,000 Genome project to really help. So that's been sequenced with short read. We're really helping them to sequence it with ultra long read to develop that data set of richness for different population, to really expand our understanding of the variation that especially in very difficult region, and evaluate which region needs to have even more insight. And as I wanted to give you just a couple of examples here, one from a publication that's not super recent, 2021, but it was done in a Chinese population, and it really showed that improving using long read sequencing really improved the sensitivity for structural variant discovery. And it actually has helped them really understanding the spectrum of structural variation in the human genome, and how rare or common they are, and actually a lot more common than people would expect. And again, is anybody doing Nanopore whole genome at scale outside of the UK? Yes, absolutely. The Emirati Genome Initiative is massive. So far, they've done 100,000 genome with Oxford Nanopore. They're really trying to do a new Arab reference genome, but they're also looking, so it's not just a population, they're also looking at variant for rare disease and cancer, like most large initiatives do, but also common complex disorder like diabetes and some of the cardiovascular disorders. So it's really an exciting program that is not in a European population, and that's really, really large. And for that, we really enable them to go into full production scale. One that I'm going to go really quickly, and I'm not going to go over that slide. I just want to go over this one, because those guys at the National Institute of Health in the US work on Alzheimer's disease and also on some other neurodegenerative disorder. And they really embraced Oxford Nanopore long read whole genome, not only because they can do haplotyping and because they can cover the difficult region, but because while they're getting the data, they're getting methylation frequency. And those guys have a lot of blood sample. They've got over 4,000 blood sample from people with Alzheimer's disease, but also they have brain sample. And so they're trying to do comparison on their brain sample and their blood sample to try and find things like biomarkers for early detection. So I'm now going to switch. Can somebody just tell me if you can still hear me? Because now I'm getting a bit panicky.
Yes.
Yes,
okay,
we can.
thank you very much. So I'm now going to switch. So the rare disease, the large cohort part is very much a research endeavor. You know, that's very much where you can try and put 2 samples per flow cell, do as many samples as you can, because you want to cover the inter-individual diversity. When it comes to rare disease, we're now approaching much more the realm of clinical settings. So Nanopore, Oxford Nanopore technology platform are research use only. We are not a diagnostic platform. However, we do have a number of groups now that are testing the platform to see if they're going to implement it as part of their diagnostic toolkit in the future. And that's where it really comes to rare disease. So rare disease are not rare. Yesterday was Rare Disease Day. They represent about 5% of the population worldwide. It's a lot of people, but 300 to 400 million people. There are over 8,000 rare disease identified. The number varies. Sometimes it's 8, sometimes it's 11,000. But it's really a lot of different disease with a lot of different phenotype. What's really important here is rare disease at 80% have a genetic origin. And out of those 80%, 70% are pediatric. They start in childhood. And those patients, those kids, their family, they can wait for 5 years, sometimes 10 years, sometimes they never get an accurate diagnosis because the current technology and method and approach do not cover everything that's needed. And a lot of people stay with never a diagnosis. It's really difficult for the family. It's also a burden economically. And whole genome sequencing, and I mean short read whole genome sequencing, is now becoming first tier test for rare and undiagnosed. So in the past, it was mostly CMA, so chromosomal microarray, and exome, very often in combination. And together in combination, they give you about 36% diagnostic yield. So people are moving to short read whole genome for the reason that it's faster. You don't need to do the pull down for the exome. You can do it more rapid. But I think what's really important here is you don't get much more diagnostic yield with short read whole genome than you get with an exome. It goes from 36 to 38, 40%. So it's just faster. It doesn't give you a better answer. And if you want to have a really good understanding of the variation across different type of variant, you need to use, at the moment, with the method used in clinical genetic, you need to use a lot of different methods. So, for example, if you want to look at SNPs or indels, sure, you can use a panel, or you can use an exome, or you can use genome, but you're going to miss the complex structural variation. You're going to struggle with copy number variant. You're going to complete miss pseudogenes. I'm thinking GBA, pharmacogenetic gene. Repeat expansion, they require using PCR or Southern. That means for the labs, it means validating lots of methods, keeping the probes up to date and validated. And every time you order more probe, you revalidate. If you're using primer, it's the same. Every time you change your primer, you need to revalidate. So it's a lot of work. And I've gone through that. The only two things I want you to remember from that slide, because the 34%, the 2,855 and the 34 are already gone over. But if you're looking at chromotripsis, for example, so those big, massive events in the genome that creates large chromosomal rearrangement, you will not be able to get them with a microarray, certainly not with short read sequencing. You might be able to see something with fish, but you're not going to have the breakpoint. The only way to understand the complexity of a rearrangement is to do it with long read and with us. And the other thing that's really important in rare disease is understanding methylation profile. And I'll have a couple of examples for that. And at the moment, the main tool used for methylation is a methylation microarray. It only covers 3%, not even, of the CPG on the whole genome. So you're really skimming the surface of methylation. You do not have a very comprehensive understanding of the methylation profile with an epic array. So when it comes to rare disease, I have two examples. I think both because they're really visual. So the first one is spinal muscular atrophy. It's a leading cause of early infant death. It's kind of treatable. It costs a fortune, but the genetic of SMA is very well understood. So, and it's also very complex. So SMA is caused by a loss of function in the SMN1 gene. It can be a deletion, it can be variant, but you lose the function. You can have somehow a little bit of functional recovery because there's an homologous gene, which is SMN2. And the more copy of SMN2 you have, if they are functional, the more recovery you're going to have. So on the slide here, we bought some sample at Cornell. And on the left, you can see in green and in green dots are the genotype from the Cornell sample that we know is the true state. And then we did our own sequencing, and it's the light blue and the dark blue column. And you can see we have full concordance with what we expect from the sample. And then on the right, we took one of the sample, and then we did the analysis of SMN1 and SMN2 haplotype. So if you look at SMN1, the red and the blue, you can see there's a deletion on both allele. There is full loss of function. And if you look at the green, the purple, and the yellow, this is SMN2. So we did the alignment. You can see there's a nice copy of SMN2 in green and in purple, and you could think there's and then in yellow you could think there's 2 copy, but actually the yellow gene has a massive deletion, so it's non-functional. So basically you've got just 1 functional copy of SMN2, and that would be really relevant if people had that in a clinical sample. The other one, because I really love that example, because it's so visual. So it's the difference between Prader-Willi syndrome and Angelman syndrome. It's caused by a mix of methylation aberration and deletion or SNPs in the promoter of the gene on chromosome 15. It's the same promoter. It's the same region. But the outcome and the phenotype from Prader-Willi and Angelman are really, really, really different. So if you look on the right, at the bottom, you've got a normal sample where you've got basically 1 methylated allele in red and 1 unmethylated in blue. In Angelman, both are unmethylated, and in Prader-Willi on the left, they're both methylated. You could say, well, it doesn't really matter. We can do that with an epicarry, or we can do that with MLPA, and we'll have that information. But the thing is, the beauty here on the slide is that you can also see if there's a microdeletion, if it's uniparental heterodisome, and therefore you have an extra information when you come to giving an answer to the molecular characterization of that sample. And I'm just going to go quickly for the sake of time. Again, if you're looking at methylation, if you're looking at an epicarry, you get about 3% of the CPGs. If you're looking at reduced representation bisulfite sequencing, it goes up. So it's like targeted sequencing for methylation. You rise to 13% of CPGs because you're really focusing on those islands. But if you use the equivalent of RBS with ONT, which is called RMS, you already have twice as much CPG covered. But I think the most important here is because currently the gold standard is bisulfite whole genome. And again, you only get about 83% of the CPG. You need to treat with bisulfite, which means you're going to damage your DNA. You're only going to get 5-HMC because if you want to have 5-HMC and you're going to have only 5-MC because if you want 5-MC and 5-HMC, you need to do 2 different treatments. If you use Oxford Nanopore whole genome sequencing, first you got way more high confidence CPG, 98%. You get information on 5-HMC and 5-MC and potentially other modifications that are relevant in your sample, whether it's blood or brain or kidney or something else. So you really get more info. But again, even if you do that for methylation, you get all of the structural variant. You get all of the SNVs and the indels. You get phasing. You can do full assembly. So in one test, you get so much richer insight. And that's the one thing I really want to remember. If there's one slide I want you to remember is this one, is that we sequence any length fragment, whether it's short or it's long. And we sequence from direct native DNA or RNA. So you get way richer insight. You get a very accurate genomic data capture for your DNA variant and their modification, the epigenetic. We do real-time data generation, and we're really scalable from an handheld device to a fairly small, compared to the other sequencer on the market, high throughput device that fits on your bench without needing to remove the windows or something to bring it in. You get way faster results in real time on the PromethION devices with the column. It's really cost effective. And again, it's very, very plug and play. The instrument I can carry our most heavy instrument. It's really easy to install in the lab. It's really easy to start working with. It's really accessible and very affordable. So that's the same. And I wanted to give you, again, a quick overview of the instrument that we have. So we do have our small handheld instrument like the MinION, so the one that's completely on the left of that slide with the laptop. That's the one everybody is very familiar with, and it's celebrating its 10 years anniversary this year. And we also have a portable PromethION that has two PromethION flow cell, again, that you either plug on the laptop or on the gridiron to get the compute. And then we go on the darker side of the slide, we go on a benchtop type of instrument with the gridiron that use the same flow cell as the MinION, and then PromethION. The PromethION device with their own compute comes in 3 different flavors, but there's the same flow cell, is the same workflow, is the same everything. The only difference is, do you want one that can run 2 flow cells and they run them independently or together, or 24 flow cell or 48 flow cell? And I think a lot of you would say, okay, that's all very good, but the data analysis is going to be a pain. So for most of the application, we work with the community to develop tools and script for data analysis. We are very aware that for applications that are going more towards clinical implementation in the future, we do need to have integration with third party. So we've announced partnership with a number of companies that do data interpretation, variant interpretation for rare disease. And those are Fabric Genomics, GenWax, Varsum, and a company in France called Sequan. So they really help you filtering and prioritizing the variant for rare disease. And we're really only at the beginning of our transformational journey. So at the moment, we're really working in research, embedded in research, or in clinical research. People, again, who are trying to see if they're going to implement or how they're going to implement Oxford Nanopore technology in the future. But we're working with, obviously, Genomics England, but we're also working with the NHS, so the National Health Services in the UK, to implement rapid sequencing in a NICU setting. As you can see on the slide, we had the Guinness World Record a couple of years ago where whole genome was done in less than 8 hours. And they actually had a massive increase of diagnostic yield compared to the standard of care, 12%. You know, if you remember that I say between exome and genomes, about 3 to 4%, between a short read whole genome and what the guys did specifically on that project, they had a 12% increase in diagnostic yield. And then there's a lot of things that come up a lot at the moment on liquid biopsy or doing DNA methylation analysis in real time in an operation, during an operation for brain tumor classification, to try and see if in the future we can really improve neurosurgical strategy in hospital. Again, people are just testing it now. We'll see how it works, but we have a lot of hope that it'll be deployed in the near future. Again, so I want to thank you all. I really am sorry I dropped my internet dropped in the middle. I hope you guys still followed, and I'm ready for question if we still have a little bit of time.
Yeah, thank you, Cora, for your wonderful presentation. So now we will go for the Q&A session. Feel free to voice out any question by unmuting your mic if you have any question. So the first question in the chat box is, could Oxford Nanopore share any projects or consortiums that are working on agriculture, forestry, fisheries project?
So, I mean, I'm really focusing on human genetics, so I'm probably not the right person, but we do have a market segment manager for plant and animal genome. So if you do have questions, just drop the NextGen scientific team or me an email, and I can put you in touch with the segment manager for plant and animal genome.
And the next question is, who is eligible for beta testing for the clinical interpretation software.
So what do you mean by the clinical interpretation software? If it's testing Safitor, Genex, Sequan, or a Fabric, we do have you can send me an email. We've got an early access user testing, but you do need to already have your own whole genome sequencing Nanopore data for the beta testing.
I see. Thank you, Cora. Is there any more question from the participants? I think there is no more question from the participants, and I think that's the end of our webinar today. Also, thank you, Cora, for the sharing, for the informative sharing, and thank you everyone for joining our webinar session today. And sorry for about the technical errors that happened in the middle of the webinar. Also, please take a moment to fill out the survey form in the chat box. And we will send you the certificate of attendance to you. Lastly, feel free to contact us at sales@nextgen.my for any further inquiries. And we hope to see you again in the next webinar.